Site-directed mutagenesis is a molecular biology technique that induces a specific point mutation such as substitution, deletion or insertion in a DNA or RNA sequence. This invaluable technique is commonly used to study the biological activity and structure of RNA, DNA, and protein due to DNA alteration, as well as inserting or removing sites of restriction enzyme or tags. Its basic principle lies in the usage of customized oligonucleotide primers that have the desired mutation onto the target sequence in the presence of a high-fidelity DNA polymerase and amplified via polymerase chain reaction, or also known as PCR. Depending on the circumstances, there are several ways to conduct site-directed mutagenesis, namely, cassette mutagenesis, conventional PCR mutagenesis, primer extension mutagenesis and inverse PCR mutagenesis. However, in this video, we will show the general procedure of a conventional PCR site-directed mutagenesis. Before beginning any experiment, always wear your personal protective equipment or PPE like disposal gloves and lab coat as part of the safety laboratory practices. Start off by turning on the laminar flow cabinet and sterilize the working table with 70% ethanol. While waiting for the laminar flow to finish setting up, bring out the PCR cloning kit from the -20 degrees Celsius freezer and place it on ice. In this example, we are using the GDP Hi-Fi DNA polymerase kit by GeneDirex. Then, place all the necessary equipment and materials into the laminar flow cabinet. On ice, assemble the PCR reaction components into a 0.2 ml PCR tube. Do note that the number of components in a cloning kit varies among manufacturers. It is important to add the reaction components in a descending order, based on their required volume, including the mutagenic primers. Based on our kit's protocol, we add 31.5 microliter of biotechnology-grade water, 5 microliter of PCR buffer and DNTP mixture, 2.5 microliter of template DNA, 2 microliter of magnesium sulfate, 1.5 microliter of mutagenic forward and reverse primers, and lastly, 1 microliter of DNA polymerase. We omit the DMSO because our target site is not a guanine and cytosine-rich target. Afterwards, vortex or quickspin the PCR reaction tube for a few seconds to ensure everything is well mixed. Place the reaction tube on ice while setting up the thermal cycler. Set up the PCR parameters according to the polymerase kit protocol. Run the thermal cycler until the reaction is complete. Once done, vortex or quickspin the reaction tube for a few seconds, and if need to be stored, keep it at -20 degrees Celsius. Next, conduct PCR cleanup on the PCR products to remove unreacted or leftover reaction components such as buffer and primers that could later interfere with the restriction enzyme's digestion. Here, we use Maturenagel's PCR cleanup kit to purify our PCR products. This will then leave us with a mixture of original and mutated DNA strands. In this final step, we are using the New England Biolabs' DPN1 kit to eliminate the original DNA strands via digestion with the help of DPN1, which is a type of restriction enzyme. Assemble the DPN1 kit components in a PCR or microcentrifuge tube on ice. Gently mix the reaction by pipetting up and down, and quickspin the mixture just for a few seconds. This is followed by incubating the tube at 37 degrees Celsius for 1 hour. After that, place the incubated tube into a water bath at 80 degrees Celsius for 20 minutes to inactivate the DPN1. Store the mutated DNA at -20 degrees Celsius until further use like cloning and sequencing. Should the site-directed mutagenesis process provide undesirable results, perform optimization upon your current protocols on areas such as primer design, primer's annealing temperature or longer DPNI digestion duration. Or it might be just some human errors like missing out on adding something into the reaction mixture or skipping a step. Now, you should have a better understanding of what a site-directed mutagenesis is, and how it is usually carried out.